Hello, everyone, and thank you for joining this World Microbiome Day event organized by the European Food Information Council, otherwise known as UFIC, and APC Microbiome Ireland. I'm your moderator today, Christina Campbell. I'm a freelance science and biotech writer who specializes in microbiome science. I am based in Victoria, Canada, though today I'm tuning in from Denver, USA, where I'm attending a probiotic science conference. And I'm excited to be here today with you to talk about microbiomes for the next hour. We have three fantastic scientific experts to learn from, and then you'll have a chance to ask them anything that's on your mind. And a reminder that you can share on social media using the hashtags hashtag World Microbiome Day or hashtag microbiomes, the number 4, and then life. So microbiomes for life. So for anyone who is not so familiar with what microbiomes are, they are collections of microorganisms, so bacteria, but also fungi or yeasts, archaea, and viruses, along with the environment that contains them. So it's like if you think of a salad made of lots of different ingredients, it must have a bowl to contain it, and a microbiome needs to have some place that contains it too. So you have microbiomes in different parts of your digestive tract, and there are microbiomes on your skin, in the soil, in your house, and many more places. This event today is called Transforming Healthier Food for a Healthier Planet. And that's because one of the most exciting things coming out of microbiome research is that they have a role in every part of our food system and what ends up on our plate. So from the soil on the farms, preparing and processing food for consumption, and ultimately sustaining our health once the food interacts with the microbiome in the digestive tract. And now that we know this, we can try to shape these microbiomes to be mutually beneficial for us humans and the planet. So to start off, we want to hear from you if you're tuning in, and we're going to do a quick poll using Slido. So for this, you have two options. You can open your web browser and go to slido.com and enter the event code, which is hashtag WMD 2023, which is on the screen, or you can simply scan the QR code and you will get to the right place. And so we're going to display the question. So very easy one first. From which country are you joining us? So just enter your country and we'll wait just a moment for the answers to come in. Ooh, lots from Ireland. I guess APC Microbiome Ireland is a popular one. Some fellow Canadians there. Wow, amazing. It keeps growing. It's wonderful. It looks truly international. All right. Yes. Oh, a few more being added. That's amazing. Okay. Thank you, everyone. So the second Slido poll. So thinking about what you know about the microbiome, how would you rate your level of knowledge right now on the microbiome? So 1 being low and 7 being high. So you can do the same thing. Okay, nice. It's looking kind of like a bell curve, right? Yeah, lots of people have a medium amount of knowledge and then it tapers off. Yeah. Excellent. So the average is about 4.2 out of 7. So we hope that by the end of this, you'll be a little higher on the scale. I think there's no way you couldn't be with these amazing experts we have to talk to you today. So yes, it is time to hear from our experts. So these are 3 people who are on the leading edge of the scientific research. And personally, I admire so much how they are working to build our knowledge about the microbiome and microbiomes little by little by making those structured observations about the microorganisms and their effects. So one note that while they are speaking, Please feel free to enter your questions into the Zoom Q&A function. We'll hear from all three of them in turn, and then we'll go into some questions for all three of them. But you can enter them anytime and we'll start to take a look. So first up, we have Dr. João Mauta of APC Microbiome Ireland presenting on the role of the microbiome in health. Dr. Mauta is a registered dietitian and senior research coordinator at the APC Microbiome University College Cork. He conducts randomized clinical trials on dietary interventions, and his main focus is to explore the relationship among dietary interventions, metabolism, and the gut microbiome. So I will hand it over to you.
Okay. Yeah. So are you hearing it? Yeah. Hopefully, yes. So thank you very much for your kind introduction, Christina. And hi, everyone. My pleasure to be here with great names in the microbiome field. I would like to thank all the organizers. My presentation is the whole of a microbiome in human health. Here are my outlines: what's microbiome and its biological function, gut microbiota and diseases, causality in humans, potential therapeutic interventions, challenges, and conclusions. Microbiome is a hot topic. The study of a human microbiome is a rapidly moving field of research, and the impact of a gut microbiome on human health is widely perceived as one of the most exciting advancements in recent years. This can be seen in the increasing number of scientific publications, as well as covers of magazines. The reason is because living in each person are trillions of microorganisms that are collectively known as the microbiome. It is estimated that the number of bacteria and human cells in our body is similar. However, we have 100 times more types of bacterial genes than human genes. Because the microbiome is a key interface between the body and the environment, these microbes can affect health in many ways, and can even affect how we respond to certain environmental substances. Most of these bacteria are located in the intestine that we call as gut microbiota. As such, it is likely that the gastrointestinal microbiome not only has the greatest impact on overall health and metabolic status of all microbes in the human body, but it also serves as a model for understanding the relationship between host microbiota interactions and disease. Animal studies have been providing clear evidence that the gut microbiome has causal role in the development of disease, such as obesity and diabetes. It was observed that in contrast to mice with a gut microbiota, germ-free animals are protected against the obesity after a consumption of a high-fat and sugar-rich diet. Additionally, the microbial colonization of adult germ-free mice produce a 6% increase in body fat content and insulin resistance. Studies with humans had reported gut microbiota differences between individuals diagnosed with diseases and healthy individuals. And these differences were frequently associated with mechanisms that significantly impact health outcomes. However, most of them are observational studies, which cannot establish causality. Human microbiota-associated rodent models are a keystone in studies with microbiome and have been used for establishing causal relationships between microbiome and host diseases. However, establishing causality is very challenging. Many published studies on human microbiota-associated rodents extrapolated the findings to make causal inferences to human diseases. Almost 100% of the papers have been reported a transfer of a pathological phenotype to recipient animals. This is extremely high and exaggerates the role of the gut microbiome in human disease. Therefore, more rigorous and critical approach for inferring causality to avoid false concepts and prevent unrealistic expectations are needed. We recently described that associations in diet studies are especially difficult to interpret as diet can affect the host health directly and independently of the microbiome. Furthermore, changes in the microbiome can just be bystanders, even if correlated, or can be the result of metabolic and immunological effects in the host, which we call reverse causality. Conversely, the effects of diet on host health can also be mediated by the gut microbiome, either by changing the composition and function of the microbiome or by over-promoting microbial biotransformation and bioactivation of dietary components and producing gut-derived metabolites. As I mentioned, the gut microbiome is at the forefront of biomedical science. But the lack of understanding of what is cause and effect is one of the major hurdles for the successful translation of microbiome research. The causal contribution of the gut microbiome to the health effects of different diets in humans is an opportunity to be investigated and one of our research lines at the APC. This will also open new perspectives and directions for gut microbiome research and applications for clinical translation, and form an important basis for the future design of microbiome target strategies, such as establishing precise microbiota-based therapeutics. Although probiotic use has been greatly popularized among the general public, and many papers have described the potential effects of probiotics on human health, clinical results are conflicting for many strains and the formulations of probiotics. Same is observed with prebiotics and dietary fibers. In this paper, it was shown that 30 grams and a high dose of inulin, a prebiotic fiber, increased LDL cholesterol and alanine aminotransferase, which is a known potential marker of liver damage. Additionally, the high dose of inulin was associated with several pro-inflammatory markers, and these effects were associated with microbiome modulation. Fibers can also act differently. Around 30 grams of arabinoxylan decrease insulin resistance and increase satiety. These results were linked to gut microbiota modulation. On the other hand, cellulose showed anti-inflammatory effect on microbiome independence. Another approach is a more comprehensive dietary intervention focused on the gut microbiome. There is substantial evidence that industrialization, mainly industrialized diet, has resulted in both an increase of chronic diseases and the gut microbiota imbalance. A study conducted by Dr. Walter confirmed the microbiome depletion when he compared the fecal microbiota of adults from non-industrialized regions with that of United States residents. The fecal microbiome of the non-industrialized cohort had higher biodiversity, and one specific strain, Limosilactobacillus reuteri, was not detectable in a single American control. Therefore, industrial lifestyle have been shown to influence microbiota composition, causing the extinction of some bacterial strains. One important strategy that can be applied to reverse this condition is what we are calling as microbiome restoration, which means provide a non-industrialized diet rich in fiber and reintroduce a species of microbes lost over the evolution of a Western diet. This study is being conducted by Aidan, a PhD student of our lab. Regarding diet, moving from Western ultra-processed diet for a diet enriched in vegetables, fruits, whole grains, and plant-based protein foods, fat fish, and low-fat and fermented dairy products are consistently recommended to decrease chronic disease. These products provide several dietary components: fiber, phytochemicals, unsaturated fatty acids, and live bacteria that benefit health through their interactions with a gut microbiota. Therefore, my conclusions are the gut microbiota clearly contributes to metabolic disorders, particularly under a diet high in sugar and low in fiber. Mechanisms and cause-effect relationships are established in animal models. However, more studies are needed in humans. A mechanistic understanding is emerging that provides a basis for therapeutic and nutritional strategies, but many concepts are not supported by a lot of evidence, and effects in human intervention studies are small. Rigorous human intervention studies are needed to determine the potential effects mediated by the gut microbiome component, establish the underlying mechanisms, and develop a microbiome target nutritional approach to address metabolic aberrancies. So I would like to thank all the foundations that support our research: APC, Microbiome, UCC, and Science Foundation Ireland. Thank you.
Thank you so much for that. Excellent. Great overview. And so what I'm taking away from that is that we know that there is a link at this point between gut microbiota and disease. Certain diseases, as you were saying, metabolic diseases such as, type 2 diabetes, obesity, and so, but we don't exactly know the mechanics of how it's happening, right? So once we do figure out that, we'll be able to figure out new therapies or even ways to shape our diets to address these conditions. So let's move to Professor Luca Coccolin. He's from University of Torino, Italy, and he will speak on the role of the microbiome in food production. Professor Coccolin is a professor of food microbiology. And not only is he a member of several international committees on food microbiology, he's also the editor-in-chief of the International Journal of Food Microbiology and has more than 350 scientific publications. So can't wait to hear from you. Take it away.
Thank you very much, Christina. I mean, let me just share with you, I'm really thrilled to be part of this event and be able to share with you the beauty of microbiome applied in the food system. I think that João just made a perfect introduction to my speech, in a sense that he really underlined how the human microbiome actually can be strongly influenced by the type of foods that we are eating. And actually, food is one of the main source of microbes for humans. And so really can work on the type of food that is part of our diet in order to modulate the gut microbiome. And especially if we get to a specific type of food that is the fermented food, then you can really understand how the gut microbiome and gut microbiota can be modulated. I'm just starting my presentation with this slide. This slide, I like it very much, and I'm always using it when I'm doing this kind of interventions in meetings, because the point that I want to underline is the colorful of the fermented foods. Fermented foods are brightly colored, and foods that are brightly colored contain compounds that are already very important for human health. I mean, there are carotenoids, there are flavonoids, there are anthocyanins. There are all compounds that have shown to be or to have a role in human health. And so if you see at the type of foods that you have in this picture, I mean, really, you can pass a lot of different products that are coming from a lot of different countries in the world. And at the end of my presentation, you will also understand how really fermented foods is a kind of heritage, an important heritage that we have in human culture that should really be maintained and should valorize. It is quite impressive from my point of view that we are talking about fermented foods as one of the most modern and recent innovations. However, if I'm going back a little bit in history, fermented foods have been around for more than almost 10,000 years. And actually, it's quite interesting to underline how fermented foods have really accompanied human beings in their evolution and in their development. Actually, the first traces and the first evidences of fermented foods are dating back at the moment humans were actually able to domesticate animals. And so through the domestication, they had the contact to their products that could or had to be conserved, had to be stored for longer periods. And by chance, by mistake, by a random event, they were actually able to transform milk into cheese. And that was really the first evidence of fermented foods. And then we go ahead with the Egyptians, and then we have Babylonians and fermented sausages. I would say that the more scientific part of the fermented food area really comes in the last couple of centuries, when we have scientists that started to work on microbes and the activity that microbes could have. We should not forget about Louis Pasteur, and actually it's quite nice to underline that the pasteurization process was actually not discovered to stabilize and to provide stable milk, but it was actually used for the pasteurization of wine, because one of the famous kings in France practically did not want to have their wine to be spoiled, and so Pasteur came with the actual pasteurization in order to stabilize it. And then you see back in the 19th century that we start to have really first biotechnological industrial application of microbes with the use of starter cultures. When we're talking about fermented foods, there is an important point that I want to make. Fermented foods are part of our life. Fermented foods are part of our culture. And so if you take a look at these pictures here that are repertoire pictures, you really understand how fermented foods had and continues to have an important role in our life. And what I like very much of these pictures is that fermented foods are actually underlining specific moments of the cycle of life. And think about the moments when grapes were picked up. Think about when olives were picked up. Those were all moments in which community got together, in which people had possibility to join and to celebrate the nature. And this is something, in my opinion, that should really be understood and maintained, because when we're talking about fermented foods, again, there is an important part of our development and our evolution. And when we're talking about fermented foods, it's also important to understand that some of the foods that probably we do not believe or we do not know are actually fermented. Here you have two examples. That is the cocoa and the coffee. Those are two products that are very important part of our diet. And also in that case, microbes are playing a role. In the case of the cocoa, a little bit longer. In the case of the coffee, a very short fermentation, but both products are really, really influenced and affected by the microbial transformations. And this is also another occasion in which I can say that when we are talking about cocoa fermentation and coffee fermentation, again, still today is a kind of social event. And so you can see in the pictures above where people are getting together in order to take out the cocoa beans from the carbohydrates, and then they start to do the fermentation. So it's still a very important social event. But when you're talking about food fermentations, here you have, from one side, the metabolic activities that are going on. On the other side, you have the map of the tube in London. You can see that, I'm not sure if you are able to spot which one is what. I mean, I can tell you that the one that you have on your left is the metabolic map of a microorganism that is living inside of our food. The one on the right is the map of the metro in London. This is just to understand, or just to make you understand, that the complexity that you have during a food fermentation inside of a microbial cell is extremely complicated. You have a lot of interactions, you have a lot of crossways. And when we are fermenting a food, we need to understand really that there is this kind of metabolic activity that is going on. So it's not a simple process, also because the number of microbes that are involved in food fermentations are really amazing. Christina, at the beginning of this event, she was talking about filamentous fungi, yeasts, bacteria, archaea. Well, this is exactly what is happening during food fermentations, because in food fermentations, we have yeasts, we have bacteria, we have filamentous fungi, that they are all contributing in order to produce foods that they have specific characteristics. And actually, you can see here that if we take in consideration one important Italian product like gorgonzola cheese, the gorgonzola cheese is the perfect example of the microbial cooperation. You have filamentous fungi that are the ones that are creating the blue color, the Penicillium roqueforti. You have Saccharomyces cerevisiae, it is a yeast, and then you have a large panel of lactic acid bacteria that are the ones that not only they are acidifying the product, but they are also producing a kind of aromas. You have already heard about the benefits of fermented foods. Here are some of the examples that are linking the fermentation with the human health. So we don't have to forget that there is also an important aspect of safety and preservation, because fermented foods are safer than the raw materials, and they have a longer shelf life. I just want to point out a little bit the story of probiotics that has been already touched also by Joao. Fermented foods and probiotics are not the same things. So there was this important paper that was published about a couple of years ago, where they really defined probiotics, fermented foods, and probiotic fermented foods. So the fermented foods that have probiotic activity are actually the ones where you have the addition of known probiotics. So let's say the concept of assuming fermented foods because they are probiotics, this is not correct. Fermented foods can have a benefit for human health, as you have heard before, but let's not let's not be mistaken about the connection between fermented foods and probiotics. And one last slide that I want to show you is about the important aspect of safety. Fermented foods, as we said, are more safe than the raw materials. However, we don't have to under-evaluate the possibility for foodborne pathogens to sneak into the product and to be maintained or to be resistant, especially if the fermentation does not succeed or is not conducted in the right way. This is, to me, very important because we need nowadays to be aware of that, especially because fermentations, as you know, are procedures that are extending more and more also at domestic level. And so we need somehow to educate the consumer in order to be able to not run into the risk of diseases that are coming from the consumption of food that are fermented not correctly. Thank you very much.
Thank you so much for that. I really love fermented foods personally. And yeah, I love that connection you make between interactions between people and interactions between microbes. So let's move to Dr. Tanja Kostic from the Austrian Institute of Technology, AIT, on the role of the microbiome in the environment and sustainability. Dr. Kostic has a PhD in food chemistry and biotechnology and is now senior research engineer at the AIT, focusing on food systems and microbiomes. She's involved in several international microbiome research projects. So I will hand it over to you.
So thank you very much for the introduction, and thank you for inviting me to participate in this great event. And thank you, and a big thanks to the speakers before me who put set the scene nicely. I did amend the title of my presentation a little bit. I said the secret rulers of the world, and I would like to show you how crucial and essential the microbiomes are in the environment, and to show you that there wouldn't be any life or any food on Earth if we wouldn't have the microbiomes. So I was also a little bit given, as I call it, the impossible task. I know I was given 10 minutes to talk about the microbiomes in the environment and just give you to a little bit set the scale. This is just now recent statistics about how many species are described at this planet, and we see all over different groups that we are in around 2 million described species, with insects really leading a sort of this bar. But when you then look at the certain estimations, is that there was a recent prediction made through different modeling that on Earth, that the Earth is a home to as many as 1 trillion of microbial species. So we are talking about here a jump from 10 to the power of 6 to 10 to the power of 12. And to make it perhaps even more visual for you is, there was also an estimation that there are about 10 million-fold more viruses in the oceans than there are stars in the universe. So we are talking about the amounts and the diversity. This is still impossible to fathom. These are only estimations. We don't know so much about them. And so I put myself at probably, I think, at the scale of 5 in the inquiry that started this talk, because there is really so much that we still don't know. But there's also so much that we do know. And what we know is sort of how it all began. So we know that this planet was created 4.5 billion years ago, and then approximately 3.5 billion years ago, a first single cells emerged, and these are archaea. And then additional 1 million years, so approximately 2.5 billion years ago, the cyanobacteria emerged. And what they did is sort of, they, as we say, invented a photosynthesis. So they started to generate the energy, and through this generation of energy, they also started to produce the oxygen. And this oxygen then accumulated in our atmosphere, and this is the baseline of the life on our planet as we know it today. So without this bacteria, we wouldn't have life on the planet as we have it. And just again, to put it into the scale, the multicellular organisms just developed like approximately 0.5 billion years ago, and the Homo sapiens is sort of the youngest one, as we know, with approximately 300,000 years ago. So as I said, the bacteria set the fundaments for the life of this earth. Bacteria are all over the place. So microorganisms and microbiomes and viruses are all over the place. And I'm just going to guide you briefly through some of the environments and show you what they do there and how we might use them to help us to, face some of the challenges that we are facing now. So starting where the life started, starting in the water and talking about the aquatic microbiome. This is still sort of an unwritten book. There is so much we don't know. There is such a diversity there that we sort of know from their genetic informations, but that we never cultivated. We still don't understand a lot of their functionalities. But what we know that aquatic microbiomes are supplying more than the half of the world's oxygen. Again, without oxygen, we don't have life on this planet. We know that many members of the aquatic microbiome produce valuable components. They produce, I have now put here fats and oils on the list, but they're also antibiotic producers in water. There are many different metabolic functions that we are now starting slowly to comprehend and start slowly to exploit and use for production of components or the food. We also know, and this is really, really important, that there are many microorganisms in the aquatic environment that can facilitate bioremediation of contaminants. If you think about oil spills, if you think about plastics in water, there's so many microorganisms that can deal with them. Not extremely fast. It won't happen from today to tomorrow, but there is a potential there that we are just learning the potential, and we are just sort of trying to figure out how to exploit it and how to make use of it. There has been in the recent year from Foundation Tara Ocean many expeditions and many efforts to sort of unlock this diversity and unlock this knowledge about the microbiome. So if anyone wants to learn more about the aquatic microbiome, I formally recommend you to go to the Foundation Tara Ocean website and look through their materials. Another big player is a soil microbiome. So soil microbiome and microbiome in general, but the soil microbiome in particular, plays a key role in nutrient cycling. So we know that they fix the nitrogen. Many of you learned in your school about the nodulating bacteria that sort of co-exhibit with the legume plants. We know that the bacteria in the soil are also able to fix methane, which is one of the greenhouse gases, and as such can sort of mitigate the greenhouse effects. We know that the soil is an important carbon sink like the water is, but they can also sort of mobilize nutrients that are bound in the soil. So for example, the phosphate solubilization, making phosphate available for the plants. The microbiomes in soil play essential role in supporting plant growth and health. And I'm going to come a little bit more to this topic in a few slides. They also produce a wide range of really valuable components. And if you just think, a lot of antibiotics that we use nowadays in the medicine have come from the organisms that were originally isolated from the soils. Similarly to water, there is also a bunch of microorganisms in the soil that are able to bioremediate contaminants, either by degrading them or helping plants to accumulate them and as such sort of remove them from the soil. This is extremely wealthy and diverse ecosystem that we are just starting to understand, and all of these interactions between the soil, the microorganisms, the climate, the plant, and everything which is affecting, this is just sort of starting to being unlocked by the community. There was also, again, a big project called Earth Microbiome that was dedicating to unlocking sort of the knowledge about the diversity of the Earth microbiome. So we learned a lot about who is there across the continents, across the ecosystems. What we still need to really learn is how they interact and how we can modulate these interactions. And then you heard a little bit about the Holobiont in the first presentation of today. So Holobiont is a human holobiont, is a human and interaction with the microbiome. But every organism on this planet, is a holobiont. So every plant, every animal, every human has its own microbiome, has many of its own microbiomes. So they have microbiomes in their gut, they have microbiomes on their skin. And similarly, the plants, they have microbiomes that are associated with the roots. They also have the microbiomes that are sort of integral plants. They live within the plants. The role of the plant microbiome, the key roles are on the one hand to provide nutrients to the plants, but they also help plants to tackle the stress situations. And what's really interesting about the plant microbiome, and something that we just recently learned, that the part of the plant microbiome is inherited through the seed. So it's like, mother plant giving something to its offspring. And then it's also actively recruited from the environment, mainly soil, but of course also through the air. The plants acquire sort of their microbiome. This is a microbiome that will then enter what Luca just told you about, that will then enter the fermentation and also play some role in the fermentation of the plant, of the food. We still don't fully understand these processes, and this is why it's important to work further on them. Animal microbiome is quite similar as what we heard about the human microbiome. It plays a key role in health development, also in aging, but also in the behavior of the animals. With animals, we also know that it can have an additional specialized functions, like, for example, if we think about the cows and the rumen and the food digestion, the microbiome of the rumen plays an essential role in degradation of this food. So it also helps cow to digest these grasses and the foods that they're eating. This microbiome can also be used to improve the plant, the health of the animals by like probiotics. But as we have fermented foods, which have many benefits, as you just heard, there's also fermented feed with its many benefits. It can be more digestible, some anti-nutrients can be removed. So there are many, many ways how we imagine that we can utilize these microbiomes that are associated with the animals. So what's really fascinating and something that we are just sort of starting to investigate is that we realized the interconnectedness of the microbiomes through the system. So what we know is that the plant microbiome is greatly affected by the soil microbiome. It's mainly recruited by the soil microbiome and it's shaped by the soil microbiome. But then if you spin and think this story further, then you have the animals that are feeding on these grasses, and the quality of the grasses that's influenced by the microbiome, but also the microbiome itself will sort of interact with this animal, and the microbiome of this animal will have direct influence of the health of the animals. But if we now think in terms of the cow and dairy, it will also have an effect on the quality and the microbiome composition of the milk. And if you then start producing milk products, especially spontaneously fermented milk products, or even milk products with starter cultures, there was a nice study done on mozzarella in Italy, and yes, all nice food studies, or most of them come from Italy, I have to admit. They show how the microbiome of the raw milk will influence the development and the quality of the mozzarella cheese. And then we are at food, and linking back to the very first presentation, the food we eat and the effect it has on the human health. But we can also think a little bit downstream, and this is still, we can use waste. We can try to use microbiomes to valorize waste, to upscale the waste, to make either food from waste, to extract the nutrients from waste. We can use also microbiomes or use them for biofuel productions. So there are many, many, many possibilities of how we can exploit on this natural wealth that we have on this planet. A look into the future, and I have already seen in the chat box some of the questions going into this direction. So what is the big question? To me, a big question is understanding interactions. We have learned in the past years a lot about the microbiome composition, but we still are at the beginning of understanding interactions within the microbiome and between the microbiome and its holobiont, and then between the microbiome, the holobiont, and the environment. And we need this knowledge to start working on the targeted applications and the modulations. What is the big potential? We see the big potential towards combating the societal challenges. The microbiomes can be used to sort of fight many of the challenges that we see in the sustainable developmental goals. We can use them to ensure food supply. We can use them towards the climate change mitigations. We can use them, as we have seen today, to promote healthy lives, not only of the humans, but also of the plants, of the animals, talking about the One Health concept. We can also use them to help us to clean the environment and to ensure the clean environment. But there are, of course, like in any science field, and please remember, microbiome is extremely young science field in comparison to some. There's still some gaps, there's still some bottlenecks, and there's still some obstacles. And ones that we have in the past years identified as a big ones are the knowledge and technology gaps. It's a young research field. We learned a lot, and we have seen, hopefully through the past, through in these presentations, how much we know, but there's many things that we don't know. And for example, one of the biggest technology gaps that we have now is that we don't know, when we are talking about the complex communities, how to sustain them together or how to preserve them together for the future. I've seen already in the Q&A box some questions about what are the hurdles, the regulatory framework, the public acceptance, the knowledge. This is everything is needed in order to push this field further and to really exploit the promise that's there. And you have seen that this presentation was sort of done on a template of the microbiome support, which is a project that just finished. And I will also like to use this opportunity to announce, and we announced it today, also in lieu of the World Microbiome Day, that the Microbiome Support Association that's currently being founded is going to keep its work by sort of engaging with stakeholders, collaborating with different stakeholders, also including the UFIC and the World Microbiome Day, that have been our partners in the year, to tackle all of these issues on different levels, to tackle different stakeholders, to share the knowledge, to disseminate the knowledge, to answer the questions, and to make sure that, you know, this wealth that we have out there is really here and can be used for all of us. And with that, I just want to sort of draw your attention to many, many resources that are out there that are available for your use. They're open access, they're free to use. We just ask, you know, acknowledgement. I would also like to thank the World Microbiome Day for the great work that they are doing over the years on spreading the word on the microbiomes and everything which is known and unknown. And I have just here taken my two favorite sort of sets of images. So one is showing how little we are using, and also a little bit about thinking that microbiomes need saving too in sense of the biodiversity. And with that, I want to thank you to your attention and thank to the organizers for this great opportunity.
Thank you so much, Dr. Kostic. And I thought it was great. Our first two speakers focused on humans and what we eat. And it's great to zoom out now to see the role of the microbiomes on the entire planet. And just, you know, I'm taking home that we're really part of nature. We're this human microbial superorganism that's part of our environment. So now it's time to take some questions, and I know that some of the panelists have been answering some by typing, but we'll try to take some verbally now. So one of the popular topics is the gut-brain axis, right? And so when we talk about a healthy diet, I'll address this one to João. So how can the gut microbiota influence mental health, and is diet a part of that, or so-called psychobiotics for health?
Thank you very much for this question. Have you ever heard the phrase butterflies in my stomach? So more and more research is revealing that the gut microbiome can influence the brain and behavior. And we have John Cryer and at APC, so he conducts several research regarding this area. And the specific gut microbes can modulate the immune system in ways that impact the brain, and also produce molecules that signal directly to neurons to regulate their activity. So we have many studies at this time that is showing that a specific strains and a specific diet, which we can say that is enriched in fiber and fermented foods, can modulate these aspects like behavior and mood.
Wonderful. So this one is for Luca. I thought it was an interesting question. You know, in ancient times, it seemed, or several centuries ago, people made fermented foods out of necessity together. But do you think in our world today, like in European capitals, we could be bringing people together to ferment food? And, you know, what would be the benefits of that?
Absolutely. I mean, I really believe that fermented foods should regain the social role that they had in the past. in a sense that, as I showed you before, I mean, when fermented foods had to be prepared, society was gathering together, was celebrating, was having feasts. So I really believe that this actually is going to be a very nice exploitation of the culture that we had in the past, because nowadays we really need to have this kind of occasions where people get together in terms of social sustainability, because we have been talking a lot about the need to engage with the citizens in order to educate them on the importance of the diet, the importance of the food, because as we heard today, foods can really be considered a very good preventive approach towards some non-communicable diseases. We are not yet there to close the gap that Tanya was talking about on how we eat and the risk that we have to certain non-communicable diseases, but for sure there is a connection between what we eat and the exposure that we have to some diseases. And this is already very common in the case of breastfed towards non-breastfed children, for instance, or babies. So I guess that this is actually a very nice idea that the participants throw there, because I mean, there are now in Europe initiatives that are moving, and fermented foods are on the radar. Let's say it is like that.
Yeah, I think it's a neat idea that microbes could not only be part of our physical health, but our social health as well. And tying it back into the gut-brain axis and mental health, right? So it's kind of multifaceted. So this one's for Tanya. Now thinking about bacteria in the environment and how they're in communities, you know, when we have like a toxic bacterium like cyanobacteria in water, you know, what can something be done to address the community and maybe reduce those kinds of threats?
We are still learning. So this is what I try to address with this knowledge gap. So we already know a lot about who is there, but learning everything which affects the community and how to push the community into the right direction, we are at the beginning. There's some really nice examples, not on a cyanobacteria, but for example, on a bioremediation, where we have a contaminated sites and we know the microorganisms that are biodegraders, where we know how we can sort of add specific nutrients that promote their functioning. So we are learning, but we need to understand these networks that Luca showed. We need to understand better these networks in the environment. They're much more complex there than in the fermented food. And we also need to understand certain redundancies and functions and how to push them and how to modulate them in the certain way. But there is a potential, definitely.
Mm-hmm. So another one for Joao here. We had a dietitian asking, you know, it sounds like dietitians make recommendations on eating fiber, plants, you know, the healthy diet. And now we have these gut microbiome companies coming in, and you can test your microbiome and they make recommendations for your diet. So is there anything that these tests can add beyond, you know, what a dietitian would say is a healthy diet?
I don't think we are at the point right now. So I think the promises are greater than what science can realistic offer. So commercial kits and maybe can tell you the kinds of microbes that live in your gut, but the results may not help you to lose weight or fend off disease. So it is impossible. So what we have nowadays, it's only association. So I would say that what we have in the market, so it is only the specific kits that we're gonna tell you the type of or the bacteria, the microbes that you have in your intestine. That's all.
Yeah, thank you. One for Luca briefly. This is a question I have, and you know, why are there geographical differences in the gut microbiome that someone has? Do you think the microbes in foods are part of this?
Wow, this is a tough question. You know what? I really believe that diversity in our gut is actually influenced is also influenced by the food that we are eating. Of course, this is not the only parameter, because we have already evidences about style of life and, all the different eating behaviors and etc. So diet for sure is one of the elements. Of course, geographic distribution is important, because in different parts of the world we have different eating habits, and so because of the access to certain foods, then we may have a difference in the type of microbes that they are in our gut. Although I can tell you that we had an experience by just looking at diet and microbial diversity in humans, just in Italy, from the north to the south. And I can tell you that even if the diet was different, the type of microbes that our volunteers had were actually the same. The functions were different, so it was not a matter of ecology setup, but it was a matter of function. So based on what we were feeding our microorganisms, the reaction or the function was different.
Excellent. And so one more here for Tanya, about the impact of urbanization. So many of us live in urban environments. Do you think we should be trying to mitigate those effects of urbanization and connecting to more environmental microbes?
Yes. There's sort of this whole green infrastructure and building and understanding, what could the sort of green buildings or effect of even your office plants have on the air microbiome. The air microbiome I didn't even touch about, because it's the blackest of all black boxes that we have. But we know that diversity matters. This was one of the motto of the World Microbiome Days a few years ago. And the more diverse environment we live in, the more diverse are the microorganisms we encounter. And it has been shown that this is beneficial for our health. And there are so great studies in especially Nordic countries, where they even sort of put the grasses in the daycare centers to see, how beneficial it sort of stimulates the development of the immune system. So definitely yes on that.
Yes, excellent. So we just have a few minutes left. I want to do one more Slido poll. So everyone who's tuning in, again, you can open your web browser, go to slido.com and enter hashtag WMD 2023, or you can just scan the QR code. And the question is, what additional aspects about the microbiome would you be interested in learning about in the future? So while you do that, I will just mention that if we did not get to your question today, you can rest assured that UFIC has taken note of it and is planning to address as many as possible in future email campaigns and on social media. So just make sure you're signed up for the list and follow them on social media. They will also give a follow-up email if you agreed to that, and send out a survey to hear, what you would like to improve for the next edition in 2024. So we'll just see some responses come in there. And also in the meantime, I wanted to mention some breaking news from my side just this week, that I've just signed on to write my next book in the Dummies series. So it'll be Gut Health for Dummies. And of course, it's called Dummies. It's called For Dummies, the series, but it's of course for smart people who are very busy and want to get straight to the point. That's the purpose of the book series. So it'll be available next March, and it's going to incorporate the latest science from experts like the ones we've heard from today. So let's take a look at the answers. Excellent. So a lot on the microbiome gut-brain axis, definitely. And some of the big diseases and conditions, that we all kind of face in our families growing older. That's excellent. So a lot of great topics there. So absolutely. I think UFIC and APC Microbiome Ireland, I can say without hesitation, are two of the best sources that I know of for, scientifically based information on microbiomes and on these topics. So I definitely recommend following them. And thank you so much for tuning in today. This concludes our World Microbiome Day event. And I want to say a big thank you to all the dedicated people at UFIC and APC Microbiome Ireland, and to our three experts today. So if you're at home, you can give them a round of applause, and we'll see you next time.